Hi. Hello everyone. My name is Xin Lei, and I am a research intern with Professor Michael Bruno, and I was under the supervision of Daria. So thank you to these two already. Thank you so much. So let's get started. The title of my presentation is Reconstructing 3D Facial Structures from Single-Nucleotide Polymorphisms. Now I know there's a lot already in the title, but I'll try to deconstruct everything slowly. And this work is still in progress, so there's going to be some stuff that's being done right at the time. So let's get started. A bit of the biological background, because I feel like most people here are AI, machine learning scientists, and so it would be good to at least get a bit of the knowledge about what is happening exactly. So our project, the main idea and the goal is to try to map from one's genes to one's facial structure. And so is there such a mapping? Well, biologically, we can say that yes. And the simple reason why is that humans have around 20,000 protein coding genes. So these are genes that code for specific proteins that can have an effect over your body. Genetically though, if we look at two humans, we're 99.9% gene identical. And so that means that 99.9% of your genes are exactly the same as the person next to you. So a bit of unifying point. But one type of variation is a single nucleotide polymorphism. So we can think of those as if we're given a pair of chromosomes, we each have one from our father and one from our mother, biological. And suppose there is a variation in one of them, we call this in one specific position within your genome. We call that a single nucleotide polymorphism, specifically a single nucleotide. So if we remember, a nucleotide is kind of a base pair and they go by ACTG. And so these determine kind of the genetic code that each person has. So is there a direct mapping from the genes to the facial structure? Well, if we look at biologically, it seems that yes. But there's also so many factors that go beyond the simple genes that we actually cannot know for certain. And that is three main factors that I listed here. So first of all, there's the epigenetic side of it. So epigenetics is simply what goes beyond the simple genetic code, which is modifications to the DNA, say DNA methylation, that can also affect gene expression. There's also histone modifications. Histones are small proteins that bind the DNA together, and these, if they're closer or further away, can affect how the gene is expressed, and therefore, even though two people can have the exact same single nucleotide polymorphism or the exact same gene at a specific position, one of them could express it and the other couldn't. Variation can also obviously be environmental. So two people can, through their diet, their sex, or no, the sex is genetic, but the diet and how they progress life, the age, obviously your facial structure will change with them. And finally, some facial structures and a lot of your traits are polygenic, which means that there are multiple genes that affect that structure or that expression. And so the actual information gain for each SNP is fairly small. Statistically, if we translate that, there are statistical analyses that have been made from genome sequencing to facial structure. But most of the predictive accuracy was in facial regions that differed the most between two ethnicities, in this case, the African and the European individuals. And so what happened is essentially they were detecting which ethnicity the person is and then outputting the average face for that ethnicity. And so, but that was a 2017 paper that relied purely on statistical basis. So is there a deep learning structure that we can learn to model this function from your genes to your facial structure? And here's the caveat that we have, is that right now, we're only working with 1,600 data points, and we know that generative models usually have, when we think about image models, they were trained on hundreds of thousands of data points. But let's still try by utilizing as much as possible prior work that either did the genetic encoding on their side or the facial encoding. And I'll get back to this a bit later. All right, so yes, other methods in terms of statistics have indeed demonstrated that there are specific SNPs that we can look at that will directly affect the face, but to a certain level of stochasticity. And so, for first intuition, right, we wanted to iterate quickly and just get an intuition about what is happening. And so, as a first baseline, we trained this very simple model that goes from the 350 SNPs that were pre-selected based on previous studies, and that went into this 300-dimensional vector that represents a flame mesh. So flame can be thought as just a parametric head model. That is a model that represents the face of facial mesh with a certain latent vector. And this vector was learned through PCA by some prior work in a lab at Max Planck Institute of Technology. Thank you to them. So they did a lot of work with disentangling the shape, the expression, and the pose, and it makes it super easy to work upon them and build this facial structure. So you give it the mesh, and they output you this 300-dimensional vector that represents the closest flame, so eigenface, to that mesh. Okay. We went through this, and we saw that there was a major problem, which is that the initial results give us a regression to the mean, which kind of is happening for multiple reasons. So first of all, there's probably very low information gain from each SNPs to the facial structure, because each SNP, actually, we can consider them as such a small change in your genome that even though some of them can drastically change, others just don't yield any information. Furthermore, we know that facial structure is highly, highly polygenic, and therefore each SNP, a lot of SNPs together need to be in a combination in a certain way to then be able to affect the facial structure. And finally, facial structure is a one-to-many task, just as I described before. So someone could have a very similar gene to you, but have completely different gene expression. So the finding is that also regression assumes this ground truth. So if we look at the previous model, we're working with this one-to-one assumption that there exists a one-to-one mapping. So instead, we should work kind of with the stochasticity of the natural environment that we have. And so can we approach this from something more generative, where we can include some of the stochasticities in the environmental factors as well as the epigenetics factors. And this is where the work in progress starts. So there's a lot of literature review that had to be done, but I'll try to summarize it as quick as possible. So we have previous generative approaches that go from the genes to the faces that use diffusion models. Their pipeline had around 10,000 data points, which is around, which is 10,000, okay, a bit less than 10 times actually, 7 times what we have. But what we learned from them is that we could leverage. So they didn't leverage any prior knowledge. They tried to just directly train their model from the SNPs to the facial structures. But now in 2025, we actually have a lot of models that can do some parts of this pipeline for us that we can leverage that would each individually train with so much more data. Specifically, I was trying to look into 3D morphable models that are not necessarily for face. So we have these morphable models that represent any 3D mesh. So we have triple SF, sparse flex, 3D shape to back set, etc. These are all extremely good reconstruction accuracies for 3D models. So the caveat of those is that their latent representation is 10,000 floating point numbers. So trying to regress those in a way that is from the 350 SNPs is like going from an extremely sparse information pool to something that's extremely dense and complicated. And therefore, these state-of-the-art reconstruction model for 3D are not necessarily the ones that we want to use. Okay, and so we are taking a look into other approaches. So we kind of lean into implicit neural representations for the face. And I'll try to go through this theory a bit, but without boring everyone. So there's a lot of work being done in implicit neural representation for diffusion models, for images, and it was mostly popularized by this paper, the NERF paper. So the idea is you kind of learn a function. Just like any machine learning model, you learn a function that maps a space A to a space B. Okay, until now, nothing special, except that the space A is a positional space. So instead of being some kind of input, the input becomes the position. So what's at this point, X, what's at this coordinate, X, Y, Z, what's at this coordinate, X, Y, Z. And the model implicitly learns to represent an image, structure, or a 3D model from the positioning that we inputted. And so we can formulate this task as something similar to f of theta given position A, and then output the distance coordinate for the face. For specifically the facial neural parametric head model that we're using, they kind of were able to boil it down to learning also to optimizing 2 latent codes. These are codes that represent each face. So for every face in our data set, we optimize a code, actually 2 codes, one for the identity, one for the expression. Then the model itself, the neuroparametric head model, takes in for each position the code of the person and tries to output, let's say, the distance from the mean mesh of the face to the expected mesh given these 2 codes. So it's like conditioning the multilayer perceptron to output the correct distance from the mean face for our face. I know this is a lot of theory, but the basic idea is that these guys were able to lower the latent representation of something that takes around 10,000 points for accurate representation. For specifically the face, they were able to lower it down to 1.3K, which is around 1,000, so 1,344 floating point numbers. The idea is that now we have something that is possible to regress and diffuse. Okay, and as for the genetic encoders, so now we have one side of the spectrum, which is the NPHM facial structure reconstruction. How about from the genetic side? So there's these guys from the Arc Institute that have done amazing work trying to basically boil down the entire genetic material of every living being into this autoencoder that has been used in everything. And specifically, we can use the first 20 layers of those to have an accurate representation for the SNPs that we have in our dataset. And this is where the work is still in progress, but we have currently this architecture where we're taking the SNPs and we're encoding them with these first 20 layers of this EVO2 model that represents, basically takes the SNPs information into this highly high information space that has been learned in this feature embedding from the first 20 layers of EVO2. Then, so given these, we can then try to we have a VAE that goes between the space, so between the manifold of the genes to the manifold of the faces, and then we try to reconstruct with the pre-trained NPHM. So our advantage is that we're trying to utilize as much as possible pre-trained models that have already taken the time to learn each individual task, and we're just mapping the manifold between each other. If that's the general idea of this. Great, so this was the major progress that we currently have, and yeah, I'm really excited to move forward. And so I wanted to thank everyone for your attention, and if you have any questions, comments, or suggestions, that would be lovely, especially because this is work in progress, so there's always an opening for improvements. Thank you.
Hey, Xin Lei, how are you doing?
I'm good, how are you?
Just a quick question on the
Yes.
validation side.
Yes.
It's possible I'm missing something here, right? Because how we all know how challenging it is to get genetic data.
Yes.
How are you able to validate your approach or methodology?
Yes. So the ultimate validation, currently the data set that is contained only has European data. The ultimate validation would be kind of to go beyond the ethnicity. And the reason I'm saying that is that we can consider European data as this bottleneck in comparison to something like Africa, which is the place where all humans came, and then there's this population bottleneck. So if it's able to generalize, that would be the ultimate validation. Now, obviously, this kind of data is extremely hard to get, and so we're currently in the process of trying to ask for other data sets and going around. But yeah, that would be the ultimate validation. And currently, elsewise, we're just doing some of the standard machine learning validation. So, make sure that your test set is well split and make sure that there's no data contamination. But obviously, this is all standard stuff. Okay.
Is there any particular evaluation metrics that you guys are focusing on?
Yeah, so for this kind of task, it's actually extremely difficult to evaluate, given that someone's face can actually, most of the best models right now, they generate a face, and then they look at which are the closest faces within the data set in comparison to the generated one, and see how many nodes it takes to jump to that correct face. And the reason why we're not working with accuracy is that accuracy right now is around 0. It's more in terms of how close can we get. And the idea, we can think of it more as a task where if the police get some genetic information, how close can we get to the actual person? And so we're not necessarily trying to reconstruct exactly, but get at least the general features would be amazing.
Makes sense.
I don't really have any questions, but first of all, how long have you been working on this?
This has been going on for around 3 months.
Still.
Yeah, it's very
Okay.
okay. Yeah.
I feel like that age is a very important factor in the facial appearance, and I have a hard time thinking you can find age with a factor in epigenetic. So how do you account for that?
Yeah, so usually, actually not usually, but in our data set, we actually have access to the age of the people and the BMI. And what previous work have done, notably this, one of them, one of the generative approaches actually includes the age and the BMI and demonstrated significant improvement in the facial reconstruction. So we kind of know that if you only have the genes, it's pretty hard to determine an age-specific face. Once you pass it in, it becomes much better. That's from previous work. But yeah, definitely you're right. It's very hard just from the genes. Yeah.
Just to follow up on that, so basically, like age, do you have access to like what kind of metadata do you have access to, right? Because there are multiple factors that can affect the face.
So age and BMI currently, and the data that we have right now.
Yeah.
Yes.
So from my understanding, this that's it. So you have the information of the demographics with the genes for that case.
Yeah.
So did you consider domain adaptation in that case or feature alignment to remove those features like the age and BMI? To remove them from the gene using domain adaptation, so you can generalize your model later on to different demographics.
Yeah, so age and BMI are not part of the genomic information. So if I'm understanding you correctly, or like, can we learn from different domains of different people, or is it?
So from the current data set you have, I think you're limited on some demographics, right?
Yes,
So
yeah,
that's
exactly.
representation.
Yeah.
One of the ways you rely on generalization, like similar to adversarial training.
Yeah.
So you remove those information that the model is biased towards
Yeah.
to be more general.
Okay, so yes, okay, that's a really good point.
Yeah.
And that's actually something that's we really need, which is something similar to contrastive learning, right?
Yeah.
Exactly. And we added a contrastive loss, but currently it didn't work for us, but we'll definitely try that afterwards. And the reason why is contrastive loss here for something that regresses with less than 1 millimeter of difference, it's not going to save it. So we thought for now it was clearly either the architecture or the data number. And I suspect it's a big combination of both. But yeah, definitely contrastive learning is going to be very important. And there was another question there.
I have two questions.
Yes.
One of this is mostly related to what has been discussed, like how do you plan to remove the bias?
Yes.
One, and then from the data set, and second one is what is your benchmarking? Like, what's the ground truth?
Yeah.
Basically.
So the bias in this data set, in terms of bias, are we talking about something like genetic bias? So we only have data from a subset of the population?
Both.
Yeah, that would require more data from different ethnicities or people from all around the world. And this kind of stuff is extremely difficult to go around asking for. And so I don't think it will be solved within the, And for the second question, can you remind me?
The benchmark.
Yes, the benchmarking. So for the benchmarking, we're currently working with something called chamfer loss. And this is actually the same types of benchmarks that we have for 3D models. So something like reconstruction of, let's say, this kind of figure, these figures, you see like all these fun objects from video games, etc. Most of these rely just on the difference between the ground truth and the reconstructed objects. This is one way of doing it. Another way is kind of to use this node approach that I mentioned earlier. And the reason why is because the chamfer loss usually is pretty bad. And if we go by accuracy, we're actually usually pretty far. And the reason why is lack of data, low information from the SNPs. So this other approach is to take in some of so you generate the face, and then you look at how close you are within your data set to the correct face. So how many faces are closer to the face you generated than the face that is correct within your data set. So obviously it is skewed towards the test set, but this is the current way.
Basically using the same data set as your benchmark.
No, you split the test set and then you train.
Yeah, but this is how you train, like a split testing, validation, such and such. But that data set is your ground truth, basically, the one that you just showed.
Yeah, that subset of the data would be the ground truth. And the best testing set would definitely be one of the different entities.
Yeah.
Because that would show ultimate generalization, right?
Okay.
Okay.